For our Cell Slam project, we decided to tastefully demonstrate vesicular transport using cupcakes. Like the trillions of cells currently in our bodies, we had to expend time and resources to produce the necessary components for vesicular transport before carrying out the process. We hope you enjoy our demonstration as much as we enjoyed making it. What you'll first observe is transport from the ER to the vesicles. To begin, the cupcakes at the bottom of the screen represent a portion of the endoplasmic reticulum, which contains cargo proteins for secretion. After SAR1 is activated by SAR1-GEF, represented by the green M&M, SAR1 then binds to the ER membrane and recruits adapter proteins like Sec23 and Sec24 to the ER membrane. We see these COP2 inner coat proteins, represented by orange M&Ms, begin to form a budding vesicle from the ER membrane. The jelly beans represent cargo proteins headed for secretion. These inner coat proteins then recruit outer coat proteins like Sec13 and Sec31, represented by the red M&Ms. These help curve the membrane and form a bud. The bud is pinched off and sealed, forming a coated vesicle. As a vesicle travels to its next destination, the Golgi, it loses its shell. Next, we'll be observing vesicle docking to the Golgi. As the vesicle approaches the Golgi, the next step in vesicular transport is docking. The cupcakes at the top of the screen represent the cis Golgi. Rab-GEFs, represented by the green jelly beans, are present on both the vesicle and Golgi membranes. These GEFs activate specific GDP-bound Rab proteins, which then recruit effector proteins once activated. Like the tethering protein represented by the graham cracker, fusion is the next step, once the vesicle is docked to the Golgi. V snares on the vesicle membrane and T snares on the Golgi membrane, represented by the marshmallow pieces, bind to each other, and as the membranes get closer, water is excluded. The outer leaflets of the membranes fuse, and NSF, represented by the blue M&M, with the help of SNAP, dissociates V snares from T snares by hydrolyzing ATP. The two membranes are now continuous, and the inner leaflets fuse, becoming one. The cargo proteins are now in the cis Golgi. What's not shown, but I will now describe, is movement from the Golgi to secretion. The cargo proteins are processed and sorted for transport in the Golgi, where protein modifications such as glycosylation, sulfation, and phosphorylation can occur. The proteins will then undergo vesicular transport again from the Golgi to the cell membrane to enter their final destination, the extracellular space. We thank you for watching, and thank your cell's vesicular transport components for allowing them to effectively secrete proteins, keeping you alive.